Thank you. Yeah, it's very nice to be here in the building. Thinking about transitioning here next month. And I would like to talk to you today about how I've been exploring the brain using single-nucleus RNA sequencing. So as you know, our brain is very diverse, the cellular landscape of the brain. So there are multiple different cell types. We can see a lot of different neuronal types that have been studied and identified over many, many years. And we know they're different both in their shape. We can see here this beautiful drawing by Naomi Habib, by their electrophysiology properties, and also by the genes that they are expressing. Also within each region in the brain, the neurons are surrounded by multiple different cell types, such as different types of glia cells and endothelial cells that also are part of this diverse cellular microenvironment of the brain. So we're interested in understanding this diversity, but this diversity also actually is a big challenge for the molecular biologists that are trying to, when we're trying to have molecular readouts in the brain. Because if we're measuring, for example, RNA using RNA sequencing, what we're basically doing is taking a piece of tissue, mashing it all up, and taking RNAs from all of these different diverse cells at once, and then getting a very complex readout that's hard to then associate it to the different types of cells that we're seeing. So the emergence of the single-cell genomics field really changes the way we were thinking about these issues, because with single-cell RNA sequencing, we can measure RNAs, thousands of RNAs simultaneously in individual cells. And this enables us, for example, to now revisit the questions of the diversity of the different cells, trying to identify new diversity of neurons and other cells in the brain that we can see. I like this analogy where we're thinking about the way we were doing the way we were doing genomics before, where we were looking at bulk sequencing, is similar to a milkshake. We're taking all the fruit and mashing them together, and now we're thinking of doing something similar to a fruit salad, where we can examine each of these fruits separately and cluster them and do fun things with them. But the main challenge when looking at single-cell sequencing, when we try to apply it to the brain, especially the human brain, is that we need to first dissociate the brain into single cells. So we take a complex tissue like the brain, and we need to dissociate single cells, which typically what we do is we would enzymatically digest the tissue and get some kind of representation of the cell body. And this is really impossible when we're thinking about an aging brain. If you want to measure fast dynamic changes in RNA, such as those induced by neuronal activity, and mainly if we want to look at also at archived brain tissues, which is most of the human brain tissue that we have available. So what I'll tell you today is about our solution for that, which is the single-nucleus RNA sequencing, and some applications of that, both to understand the cellular diversity in the mouse and the human brain, and also to look at inter-individual diversity, both in the cortex and in the deep white matter. So the basic idea was very simple, was if the enzymatic digestion to extract single cells is complicated, there were actually many different methods to extract nuclei directly from the tissue. And this has been done, for example, in Chris's Walsh's lab long before I even started my postdoc. But there were two kind of emerging questions about the idea of doing single-nucleus RNA sequencing. Is it first possible to extract RNA from the small amount of RNA in the nucleus and perform RNA sequencing of that amount of RNA? And second, we wanted to better know what is even the information that we can get from RNA only in the nucleus, which is the most immediate RNA that's being transcribed, compared to RNA from cells. So since we started this work, there's actually many works by different groups and by our group as well that have been, I would say, established already the idea that indeed we can do in a very robust way single-nucleus RNA sequencing, And we can use the RNA in the nucleus itself to identify the cell types, new diversity of subtypes of cells, and also dynamic cell states. So this has been very exciting, and today I'll go through some of our results and also more novel and unpublished results. So first of all, just to kind of set things straight, we wanted to show you just a quick comparison between nuclei and cellular RNA. So here is actually RNA that we took from a cell line, where we can see that the number of genes we can detect in the single nuclei in cells is quite different because of the different amount of RNAs. However, when we correlate the expression patterns averaged over single nuclei and single cells, we see that we get a very highly correlated expression, with, of course, some differences, but overall we see that we do capture the same population of RNAs. We also get high quality libraries that most of the reads map very high confidently to the genome, but there is a very big difference in what is the context of these reads, where in single cell RNA sequencing we mainly get exonic reads from mature RNAs, versus in the nucleus we get almost 50% of the reads are intronic, because what we're capturing is a lot of the pre-mRNAs as well, which is interesting on its own, but it's also important to understand that. So actually, most of the talk, I'm mainly doing the analysis on the exonic reads, just kind of a small comment, but in the more preliminary data I'll show at the end, we're integrating these intronic reads into our models, and it significantly improves our analysis. So the first method we developed was single nuclei RNA sequencing, which was relatively low throughput because it was based on fact sorting cells into 96 well plates, and then performing RNA sequencing, making an RNA sequencing library on each of these individual nuclei that were captured in the individual wells on this plate. And this it's a highly robust protocol where we capture full-length mRNA sequencing. And we showed that we can generate these libraries both on fresh, frozen tissue, or even lightly fixed tissue. And what we saw is that we can definitely, based on this unbiased sequencing, we can then cluster the cells and embed them into two-dimensional. So I will quickly walk you through this graph, because we'll see a lot of more such graphs in the next few slides. So what we see here in this graph is an embedding of a huge matrix of measuring thousands of genes in individual cells into two dimensions, where every single dot is one individual nuclei, and the closer the dots are in the plot, it means also that on the transcriptional level, these two cells are similar. And we then color this graph based on a graph clustering algorithm. And what we see is we indeed identify these highly distinct groups of cells that map one-to-one to the different cell types in the brain. So we can also look at this graph this way, where we actually color the dots based on an expression level of a known marker gene. So here we can see a marker gene of granule neurons in the data gyrus, which nicely maps this cluster. And this is a way to annotate these cells. We then go back to their expression, to their pattern in their brain, to their spatial position, to annotate and understand what are the cells that we captured. So, of course, capturing the known cells in the brain wasn't the most exciting thing about our work. I would just like to highlight two things before I move forward, is we were able to actually go deeper into some of these neuronal cell types and understand additional diversity that we didn't appreciate before. So specifically in the pyramidal neurons in the CA1 and in the CA3 brain region, we found that they're actually not, even though morphologically they look the same, they actually are composed of different sets of neurons that are each transcriptionally very different from each other. We then wanted to understand, what does this transcriptional diversity hold? And one of the hypotheses was that each of these populations will actually be sitting on different subregions of the CA, which we know have different connectomics and electrophysiology regions. So indeed, we used known landmark marker genes that are from in situ hybridization data to computationally predict the position of each of these subclusters in the CA region. And what we find is we could map these transcriptionally diverged sets of neurons into spatially distinct regions, meaning that we're actually extending the anatomical resolution in which we can now understand the hippocampus, or the CA region specifically. But taking a closer look at these clusters and their mapping really highlighted one very important feature, is that these sets are actually not binary, but rather what we find is a gradual transition in expression profiles along the CA region, which I think is actually probably at some resolution, it's a much better description of the neuronal diversity, which is not only built of these discrete sets of neurons. We could actually very nicely also identify differences in cell states, dynamic cell states, and not only in these constant cell subtypes. Here, what I'm showing is that in our DeepSeq method, we actually coupled pulse labeling by EDU of dividing cells with our sNuc-Seq method in order to capture rare dividing cells in the adult brain. And we could then embed them into 2D and see that they nicely align along a maturation trajectory of the neurons. And this way, we could capture dynamic transitions in gene expression along this maturation trajectory. And this shows that in the nuclei itself, we can very easily capture also changes in very small resolution along states of cells. So overall, what I hope I convinced you is that single-nuclei RNA sequencing is a really good method to use when we're trying to understand at the single-cell resolution both cellular diversity and different cell types, and also dynamic changes in RNA. So what I next wanted to do was actually expand this and look more globally at the mouse and human brains in better resolution. But then we faced another challenge, which is the challenge of the scale. So if we want to scale, really understand the diversity of cells, we need to look at more than one state. We need to look at different ages, different disease states. And also when we're scaling up to humans, then the diversity between individuals is very large, and also the brain is just bigger. So we coupled our single-nuclei RNA sequencing method with a droplet microfluidic device that was developed before us called DroNc-seq, and where in each drop, we capture now the cells in individual droplets and barcode them in order to enable sequencing of thousands of genes at very high paced and relatively low cost. And this enabled us this important scale. Just to note that since we published this work, few other microfluidic-based methods are now available, so people can kind of choose the best method that works for them, which is amazing. And the first application that we did was, again, going to the mouse hippocampus, and also we took a prefrontal cortex region and used and clustered it in the same method I described before. And what we see that, again, we capture all the different cell types in the brain. This time we have many more cells, so our confidence level in the classification is much higher. We can easily identify subtypes of cells. Even here in this big clustering, we already see different subtypes of excitatory neurons in the prefrontal cortex, for example. I just wanted to highlight this one example to show that we're actually capturing not only different cells along the different layers of the cortex, but also, for example, in this small cluster, we find a small cluster of cells that are different in their state and not in their cell type, because they all express set of known immediate early genes, meaning we captured a set of neurons that were recently activated. We then did the same thing about going to the human brain. So we took, again, hippocampus and prefrontal cortex regions from archived post-mortem human brain. And I was really excited, actually, to get these results, because these tissues were sitting in the freezer for between 3 to 5 years before we actually did the analysis. And so we weren't sure about how well could we capture these slightly degrading tissues. And again, we can find all the different types of cells also here. So we have astrocytes, oligodendrocytes, inhibitory neurons, and excitatory neurons, etc. And we next wanted to compare how well does the expression patterns of these cells correlate. So what we see so far is both in the mouse and the brain, as we knew very well, we have the same type of cells, of neurons, and glia cells. We wanted to see if their expression profile, their cell type specific expression profile is correlated. And what we find is that indeed when we correlate the expression profiles, both of the mouse DroNc-seq to our previous sNuc-Seq data and the human DroNc-seq to the sNuc-Seq data, we see a very nice correlation, where of course the correlation goes down a little bit in the human cells, but still we see a very high correlation, meaning that the cell identity is maintained. So there are many differences that we could look at, but it's a mixture of technical and other confining factors. So what we decided to focus on for the comparison is, first of all, go deeper into the subtype level of the cells and ask, are these also conserved? And what we see is that we can, for example, take the GABAergic neuron cluster and identify additional subclustering of these cells. You can even go deeper. Each of these subclusters goes with very known marker genes, such as for valbumine neurons and somatostatin, etc. And we can then take our clusters of GABAergic neurons and build a linear classifier to see how well does these human GABAergic subclusters capture previously identified subsets of GABAergic neurons that were identified in the mouse visual cortex or the mouse hippocampus. And what we see is we see quite a clear, almost one-to-one mapping between our clusters and the previously identified clusters, which shows that also at the subtype level, we still see a very high conservation, also with the full transcriptional profiles. Another aspect that we could actually compare was looking at disease-associated genes. So here what I did is I chose different genes that are known to be associated with Alzheimer's, depression, and schizophrenia, and I look at their expression pattern along the different cell types from our data set. So we can now associate the disease gene to a specific cell type. So there are two interesting things that emerge here. The first is that we see that indeed all of these diseases, even a disease like Alzheimer's that we know is affecting the neurons, has all the cellular microenvironment takes a really important role in the progression of the disease. Because we see that not only neuronal cells are indicated by these disease genes, but many other different cells, such as microglia cells and astrocytes and Alzheimer's, for example. The second thing is that we see different diseases have a different pattern across disease states. So what happens when we compare these disease-associated genes between mouse and human, we see that overall the patterns are quite similar, but there are specific genes that have a different cell type associated pattern, which could also explain why the mouse models that we have don't always recapitulate nicely the disease. So overall, what I showed you here so far is by looking at the cellular, by using our DroNc-seq technology, we could look at the cellular diversity both in the mouse and the human and try to compare them both at the subtype level and at the expression pattern level. And what we're doing right now is we're expanding this view into looking deeper into the individual diversity within the humans, because this is another very important aspect that we didn't have enough power to do in the first work. And I'm doing it by looking both at the cortex, but also diving into this incredibly interesting region of the deep white matter that I'm glad that it was brought up in the previous talk, because we see that by many emerging evidence that showing that the deep white matter is also a major driver of disease states. And we wanted to better understand not the cells themselves that sit in the deep white matter. So what we see here is in our new data set, we profiled 24 aging individuals, different sex and disease states, and we looked both at the frontal cortex and the deep white matter. So the first thing that we saw is that indeed we have a very unique cellular landscape between these two regions as expected. We could see that even from this shape of the clustering, where the deep white matter is dominated by a huge set of oligodendrocytes. And in addition to that, of course, we find a large set of microglia cells and astrocytes, and even some neurons, endothelial cells, and oligodendrocyte precursor cells. In the frontal cortex, we again kind of recapitulated the same cellular diversity we found before, only this time we have a much more rich data set, which enabled us to actually look deeper now into the diversity of also the non-neuronal cells, finding incredible diversity in both the microglia cells, the astrocyte, endothelial, and other cell types, oligodendrocytes as well, which was much larger than what was previously reported in mice, for example. One interesting thing is that when we co-cluster these non-neuronal cells between the deep white matter and the cortical regions, we see some very dramatic differences. Here I'm coloring the graph by the region, and especially the oligodendrocyte cells. There's a huge region that's, I don't know if it's my timer, that's specific to the deep white matter or the medial frontal cortex. So this shows that not only by looking at this new region, we're actually finding a whole new set of glia cells that are very distinct from what we were seeing before in the cortex, which was our main focus up to now. But going back to the question of the inter-individual diversity, here I'm looking at the same graph, and each dot here is an individual. We only have 16 samples actually on this graph. And the first thing that came that I saw was that, first of all, we see a lot of diversity, but first, the individual first clusters by the cell type. Okay, so the cell type identity is so strong and so conserved that we can take people that are extremely different from each other, but we're still, the most strong information that we have from our RNA is their cell identity. But then when we look deeper into these cells, we see incredible diversity. But this diversity is not consistent between different cell types. So for example, astrocytes compared to excitatory neurons are much more diverse between individuals. And this actually goes nicely with what we know about the astrocytes as sensing the environment, both the internal environment and the external environment. It also highlights the importance of astrocytes for disease progressions and other glia cells as well. Actually, one of the major drivers here of this diversity is, of course, the sex differences, because I have both females and males in this process. So we can see here that, as expected, we're different from each other. These differences are not only derived from like X and Y related genes. And we find many genes that are different from that, and moreover, they're very cell type specific. So we can find, for example, an inflammation-induced genes that's highly enriched in females and microglia cells, but not in any other cell type in the brain. And we can find an interferon-induced genes in endothelial cells of females, and that's not so enriched in males. That also is, we cannot see that when we look at other cell types in the brain. So the combination of both these things shows us that in order to understand disease, we want to look both at the cell type level and add this individual diversity into it. And of course, we see both the importance, again, of the entire cellular microenvironment in disease progression, and here specifically in inflammatory response. So this is actually my focus in my lab, where I'm trying to understand how the different cells in the microenvironments are interacting between them and promoting disease progression, and how they're involved both in the degeneration process and also in the repair and regeneration. And we already started doing that by using our single-nucleus RNA sequencing method until another method will take over our lab, looking along disease progression at different stages and trying to model these interesting interactions between cell and cell type specific changes. In our initial data set, both in mouse and human, points that we do have incredible changes along disease progression that are very specific to different cell types. So this is overall what I had time to talk with you today, both starting from the single nuclear RNA sequencing, going to the comparison between mouse and human cells, and then focusing more on the inter-individual diversity between, within the human population. So I want to just end by thanking, first of all, the donors and their families, the brain donors that really enable everything that we're doing here. Secondly, my postdoc advisors, Feng Zhang and Aviv Regeb, who enabled me to do all this work. My colleagues, Yingqing, who is a co-author in the first paper I showed you today, along with Lukasz and Matthias, Inbal and Oni, or the co-authors of the DroNc-seq paper, along with a lot of help along the way. All the more preliminary data I showed you is in collaboration with Phil DeJager and Kristen McCabe and David Bennett, and these are my other collaborators and some funding. I just want to end by thanking LSEC for accepting me to be part of this amazing community, and I'm very much looking forward for next month. Thank you.
Did you look into correlations between the different expressions in sexes and different paths of disease advancement?
So, if I understand, you're asking if there's a correlation between the sex and the disease progression? Yeah, this is actually, so this data is very preliminary, but I can tell you from my preliminary analysis that it's actually a very big challenge, because what we find is that we have both sex differences within a given cell type. We have a very dominant sex difference, but it does correlate with the state of the disease. And this is, of course, important to understand, because it means we need to study the disease differently when we're thinking about females and males, and even when we're thinking about developing therapeutics, they might change between them. But currently, from wearing my computational hat for a second, it actually really complicates the analysis. So trying to find better, clever ways to kind of pure the information from there.
Yeah. Beautiful. I wonder if you have also information on small, like microRNA in these nuclei, and moreover, if I may ask, on can you typing the cell by alternative splicing? You mentioned the intron domination in your preparation.
Yeah, great questions. So about the microRNA, it's interesting because in the nuclear RNA, I didn't mention that, but we have an enrichment for long non-coding RNAs that are localized in the nuclei. For the microRNAs, usually in mRNA sequencing, these are poly-A-based RNA. We miss them. But actually in the nucleus, we do capture the pre-microRNA before it's being kind of processed in the cytoplasm. So we do see it, but it's, most of them are kind of at a low level that I was a bit afraid to report to, but we are looking into it. And it's something that's, thanks for the reminder, it's definitely worth it. Regarding the splicing, that's very exciting. Right now, I didn't mention that, but our new data sets that are large scale and have gazillion cells are actually 3' based. So we're missing some of this. What we can see is like 3' alternative sites. Exactly. And that's interesting. In our more low throughput method, we had the full length RNA, and I did do this analysis. And I found surprisingly pretty low amounts of like splice specific RNAs that are changing between cells. I think part of it is actually because of the noise. So single cell RNA sequencing is relatively very noisy data set with a lot of dropouts. And what we're going to now is moving from we're trying to analyze the splicing by first grouping the cells and then averaging over them. So using the averaging to kind of deal with the noise that we're handling. So, yeah.
Yeah, this was wonderful, and along the same line of question, Hi.
Oh, okay. I didn't know who to look at.
Along the same line of questioning, women do show age-related increases in inflammatory markers much more than men. So you might need to do the correlations at several dimensions, both age, stage of disease, of course, sex-related, and when women get sick, they deteriorate much faster. So that comparison would be challenging in view of the conflicting theories of the role of inflammation in Alzheimer's.
Right.
So.
Yeah, but it's also, I was actually excited to find the first thing that pop out of the analysis to see these inflammatory genes, because it might underlie what you just highlighted, the differences we already know in inflammation-related changes in the aging brain between males and females, and might actually be the mechanism we're looking for. So, but yeah, still dealing with the noise.
Okay, so I want to thank Naomi, and actually now we're going to